if you think, answer yes or no if you feel that you have a good understanding of what serology is. Okay. So it looks like I'm getting a lot of yeses and some nos, but it's maybe like 30, 30, 70 or something like that. And so I'll ask another question. How many people, if you are here more for a if you are more interested in there will be two answers here, either technical or epidemiological. So T or E if you want to do that. If you're here more because you want to know about the technical aspects of serology, right, technical, and if you're here more because you want to know more about the application of serology and in epidemiology, then add E. This is a very, very not scientific way to get this information, but it works. Okay, perfect. So it seems like there's a pretty good mix. I just wanted to get a feel for that. So I'm going to start by talking a little bit about what has happened sort of thus far to get us to a point in this epidemic where we really need, where we're moving into this era of serology and why it's been sort of all in the news recently, and why it's just so important. And I'll try to dispel some myths for a few minutes about sort of what has been said about serology so far, and maybe where it will go, what it will do, what it won't be able to accomplish, and where we need to go with it. Thus far in the epidemic, you just heard all about the epidemiology of the epidemic and a lot of nice modeling work by members of CCDD and elsewhere that sort of discusses the overall epidemic and its dynamics. But one of the major features that's used to drive a lot of those models is testing. And thus far into the epidemic, and everyone's probably heard about testing at this point, it's been on the cover of every major media outlet. And it's, you know, everyone probably knows more than they ever wanted to about swabs and problems with swabs. But swabbing and doing the PCR-based testing, which is actual detection of the virus, is very important. As this epidemic got going, it's an important clinical tool to know who's infected and who's not. And it's also then, of course, those data are used to be placed into epidemic models and dynamic models like the ones you just heard about to understand what is the epidemic doing. And what that means, though, is that in order to build those models, we really need to have good understanding of what the data actually means. And part of that might be how many people are actually infected, what's the positivity rate of these tests versus the numbers of positives versus negatives. Questions like that come up all the time. But viral testing is really a clinical tool in many ways. The virus, when it goes into somebody and is detectable in a person, it's detectable for a short window of time relative to, you know, on a time scale of months or years. People only carry the virus for a week or two. And so that means for any individual, you have a very short amount of time to capture that virus in them in order to be able to tell somebody, yes, you have been exposed to this virus and you have been infected. And all of the data that we've been using for the most part throughout this epidemic has been data, at least from a biological perspective, has been data that's based on detection of the virus itself. That, of course, runs into problems. For example, when testing isn't available. If you miss the whole beginning of an epidemic, like we in many ways did here in the United States, then you spend the rest of your time trying to catch up to the epidemic using testing. And you run the risk of missing many, many people during that process. So a lot of people who might have been exposed throughout March, but in areas of the United States, for example, where testing was not yet available, you'll end up running into big problems because you won't be able to necessarily know how many people you missed during that portion when testing was getting ramped up. In addition, as testing is ramping up, you have the virus is growing exponentially as you're just kind of scrambling to get testing going. So because of that, a lot of the testing data that we have from the virus is inaccurate. And we're not even sure in what ways it's inaccurate. We can't just say we missed 80% of cases or 50%. We can, we have to, we don't know exactly how many cases we actually missed, and we have trouble scaling our models appropriately. So this brings us to serology. So PCR, like I said, looks for the virus. Serology, on the other hand, looks for something much more long-term than the virus. And it essentially is utilizing the immunological memory that our bodies naturally create every time we are infected with a pathogen. Our bodies essentially have a record of immunity of those pathogen exposures, and it imprints in our system a recording, and that recording in this case are antibodies. And so we can actually use those antibodies to detect whether or not somebody's been exposed in the past, and those and different types of antibodies we can look at will give us different windows into somebody's previous exposure history. Some of it, if we use an antibody called IgM, that's an antibody that's very short-lived, relatively speaking, and it might only persist for a few weeks to a few months before it disappears again, but it will usually come up around a week after infection, maybe 2 weeks after infection. So IgM antibodies aren't the greatest for diagnostics. You'd rather use a viral detection if you're truly trying to find somebody when they're infected. But you have a little bit more lag. You have a bit more leeway to actually detect somebody over the next month or so to be able to say, we believe that you have been infected in the last month. And we can do that with IgM. So that's actually a very powerful tool to do near real-time surveillance of infections using that antibody. IgG, on the other hand, is a very different antibody, and it is also raised in response to an infection. It's our immune system's way of recording that infection happened. And of course, these actually have utility beyond just giving us a window into somebody's infectious status or infected status. They also, of course, help to potentially neutralize and protect you from infections in the future. But in this case, in serology, the study of what's in the serum and the blood, we look for antibodies as a way to measure whether somebody has had an infection in the past. IgG doesn't give us a good window into knowing exactly how far in the past we're looking. If somebody has an IgG titer against measles right now, maybe that's because you got measles last year. Maybe it's because you had measles 50 years ago, and it can be difficult to tell. But in an emerging outbreak like this, we know that the window of time is pretty short. And so we know in this case that most likely everyone who has IgG antibodies against this virus got it sometime since December. And we can use that to understand in different settings what has been the overall cumulative incidence of this virus, and it can start filling in some of those gaps. The gaps that we missed with the PCR testing for the virus itself, we can actually start filling in and figuring out how much of the cases we missed by looking at antibody tests. And so that's one of the major reasons that in a public health sphere, we use antibody tests all the time in order to know, in this case, where are we in this epidemic curve? And but the downfall, if you're using IgG, is that you're kind of 3 weeks behind the curve. So if you're measuring people with IgG antibody titers and serology looking for those, that usually gets raised about 3 weeks post-infection. So you can't count on it to be sort of an up-to-date window into the epidemic, but you can make pretty good projections based on it, because in general, your power, if you're looking at antibodies against IgG, will be greater than your power if you're looking at virus PCR, just because you'll be able to capture a much greater number of people to understand how many people have had it. So we really are looking at antibodies now, and IgG antibodies, and you've heard all about serology, in part because there were just so many blunders with getting the PCR tests up and running that now everyone is clamoring to know where we are in the infection, in the epidemic. And the reason that this is important, the reason that serology and understanding the overall prevalence of this pathogen is just so important, is because it will determine in many ways what steps we end up taking next. Right now, we have in across the world less than, I believe, I haven't looked today or yesterday, but I believe it's under 2 million reported cases of coronavirus across the globe, and many hundreds of thousands in the United States. But that's reported cases based on PCR. If we use that data and we derive estimates of how many people have been infected based on that data, then it makes our infection it makes the mortality rate of the virus look very high, which it might be. But if we start doing serology and recognize that we actually missed 90% or more of all of the cases that have happened in this country, then it really changes our view of many, many things. First and foremost, it changes our view of how many people have been infected and remain susceptible to this infection. But it also changes our view on the actual biology and pathogenicity of this virus. So just being able to know the actual cumulative incidence of people getting this infection in the past few months will determine and change how we're looking at this virus. If we find that in the United States, for example, 30 million people have had this virus by looking at their serological status, then that will really change the way that we interpret the individual level risk features of the virus to cause death. And it will be quite a bit lower than what our current estimates suggest, which is usually around 1 or 2%. If we find that we were an order of magnitude off or more, then maybe it's going to be 0.1%, for example. And so this is one of the major reasons that we really are kind of clamoring to get serology tests and assays available to the masses. There's another big reason, and the other major reason that serology has become so important is because we need to figure out how to get the economy running again. So part of that is the prevalence and the public health aspects that I was just referring to. If we realize that the individual level risk of getting infected is lower, then we can start to think about other ways to navigate opening the economy. On the other hand, we want to also use it on an individual level basis. Right now, we have huge numbers of people in our hospital systems who are not able to work. And this is because they have recently been infected, or because they're at very high risk for having been infected. And we worry that we maybe have missed the window of finding out whether or not they were. And so one of the great things would be if we had some way to know if people could be safe to go back to work, or if people could be safe to start working with elderly individuals and nursing home patients, for example, the most high-risk patients among us. For people to really start working with those individuals, we want to make sure that they're not infected. And one of the best ways to do that is further to make sure, or to try to understand if they're actually immune to being infected, if they can't get infected. And so, so one of the major pieces that we're trying to do is figure out how can we use antibody testing to understand whether or not somebody is immune, and what does a positive antibody test mean clinically. So this is sort of a phase of this epidemic that we're getting into now. We've been trying to understand the epidemiology of the virus. We've been trying to understand the PCR-based results of the virus and what the dynamics of the virus and the kinetics of the virus look like in the body. And now we're trying to understand, it's really moving into a phase of trying to understand the immunological response to the virus. And one of the big problems and one of the misconceptions that you may have heard on the news or elsewhere is that people and sort of the CEOs of corporations and sort of anyone out there who owns a business really wants their employees to work again. And this is maybe most pronounced in hospital settings, which are currently being slammed by patients, and the physicians and nurses have to be away from the hospital because they're infected. So how can we get to a point where we know that somebody's safe to go back? Serological studies have to be done. We can't just say that somebody has an antibody titer and assume that they are protected from reinfection and that they are unable to go and give an infection to somebody else. And as much as we would love to be able to assume that, there's many examples where just because somebody has an antibody in their blood that's detectable and has evidence of having been exposed, there are many times when people can actually still become reinfected. One of the most obvious cases is certainly influenza, where we know that it's not a fully sterilizing infection. So even though people have antibodies to flu, they can still go on and get flu again, sometimes even the same strain of flu. And so we know that this can be a problem. And this is sort of the era that we're in now, or the months that we're going to see ahead are going to be a lot of clinical studies where we actually are taking people and we're testing them serologically to know, do they have antibodies or not? Or we're testing them to say, do they have virus in them, and then following them in time. As we follow them in time, we'll be able to measure how many antibodies do they have in their blood, what titer, so the number of antibodies, we call that a titer. And we can essentially count the numbers of antibodies or quantify the titer of potentially protective antibodies they have, and try to figure out, are there certain levels of antibody responses that people have that will prevent them from going on and becoming reinfected. There's a chance we might find that anyone, at least for months or a year or so, anyone that has antibodies will be protected from reinfection and will not be able to reinfect other people. There is that chance, but we might more reasonably expect to find that you have to have antibodies above some threshold in order to go and gain protection from reinfection. And so many of these you might have seen pictures of the antibody tests that have been sort of they've been built in China and Korea in very large quantities and sort of shipped around the world. They look a little bit like a thermometer or like a pregnancy test. And these are point of care antibody tests that can be useful, but we've also found that they're sometimes inaccurate. But what they also don't do is they don't give a quantitative value for the most part. And so we need to develop the tools and the tests that actually give us a quantitative number for how many antibodies somebody has in order to understand, in order to figure out how to interpret these results. And so here at Harvard and elsewhere all across the world, people are now enrolling in studies in order to participate in these. So somebody who knows that they've been infected can enroll in a study, and they can get their antibody titers drawn from a blood sample or just a blood prick, a finger prick. We can then go and test their antibodies and then follow them in time forward to say, do these people get reinfected again? And hopefully we'll find that none of them do, but likely we'll find that at least some do get reinfected. And that will allow us to start creating what we call correlates of protection or thresholds of protection. And we would be able to say to somebody ideally with good confidence, your antibody titer is 400 international units. And that means that you're safe as far as we're concerned, at least for the next 6 months from reinfection. So those are the types of values and studies that we're going to have to do moving forward to really understand this information before we can actually, with any good confidence, tell the owner of a company that he can or she can have their employees come back to work, for example. And so despite there being this amazing fervor to get antibody testing performed on everyone, as clinicians and as scientists and as researchers, we really are, I think, cautioning that this needs to be slowed down a little bit. The brakes have to be put on just a bit in order to give some time for these trials to move forward, to understand how we should be interpreting these values. And so I'm going to just mention a couple of additional pieces to this, and that's kind of looking at the technical aspects. So you might hear the word ELISA coming up in the news a lot. I think a lot of people are becoming familiar with words they've never known before. ELISA is a type of technology, laboratory tool that we use to measure antibodies. And it's a pretty tried and true and sort of trustworthy method to measure antibodies. However, it doesn't measure the functional response of antibodies. It only is telling us, are antibodies existing? And this is important to recognize because you could have an antibody if you consider yourself a pathogen, and an antibody is, I don't know, to take your phone or your hand or something. If I'm a pathogen and this is an antibody and it hits my shoulder, it's not going to do anything to me. That's not going to neutralize me as a virus. But if I'm a virus and this is an antibody and it covers my face and squishes it, then it's really going to maybe neutralize me. And so it really matters where these viruses bind. Where the antibodies bind on the virus. And that's going to essentially impact how important or the role of those antibodies in people. And so, but an ELISA-based assay only measures how many antibodies you might have on you, but not where they are. So there's another type of antibody, the test that we call neutralizing antibody titers and or neutralizing assays. And this is actually an indirect way to look for antibodies. And we don't actually quantify the antibodies themselves. We quantify the functional effect of those antibodies to prevent growth of a virus in a Petri dish. And so you can essentially take a bunch of Petri dishes and put virus in each one and drop different dilutions of somebody's blood into each of those and see what is the greatest dilution that you can go to while still neutralizing the virus so that it can't grow in that Petri dish. So if you do one dilution from like do 50% serum and 50% water, for example, then you will, that would be a pretty low titer. If you can only dilute it a little bit before the virus stops being neutralized, and that's a very low neutralizing titer, and that person might not be protected. However, if you can make 1,000 dilutions and still get protective efficacy and still have that blood sample that's now been diluted many, many times, preventing the virus from growing, then that's a very high titer that person must have, and a very high neutralizing titer, and they'd likely be protected. So those are two extreme examples. It's usually not either of those extremes. It's usually something in the middle. But the notion of neutralizing antibody titers is very important as you start to interpret how to, or as you start to think about interpreting these serological studies. I think it's just important to remember that ELISAs and the majority of tests we do to measure antibodies are not functional. And so there might be some people who have lots of antibodies but can't kill the virus. And there might be some people who have only one really good antibody, and it's at a pretty high titer, and it could actually do a good job at neutralizing a virus. So they're getting at that. Sometimes the ELISA tests don't tell the whole story. So in some ways, our gold standard for serology are these neutralizing assays. So it's 12:50, and I thought I would just stop there. I gave sort of a blabbering whirlwind tour of that, and I thought I'd just take some questions, which looks like some people have already asked. So one question says, what's the relationship between antibody level and shedding? How long will a patient be infectious after the symptom onset based on the knowledge we have known so far? So this is a really great question. I think I kind of addressed it during the last few minutes, but we don't yet know what is the relationship between antibody levels and shedding. And these are the studies that we really have to start doing. We don't know if they'll prevent shedding at all. We assume that this virus will be just like many other viruses that infect humans, and they will prevent shedding and they'll prevent infection to at least a large degree. But we don't yet have the data. And so the studies that will be happening in the next month or so, or getting started right now across the world, are really going to help us understand that. So what is a better target, IgM or IgG, to understand community transmission if both can't be evaluated? So if you're trying to choose between an IgM-based assay, if it was me, I think there's philosophical differences on both. If it was me and I was having to choose whether I was going to use IgM or IgG, I think if I thought that I was at the beginning of an epidemic, then maybe IgM is okay. If you really want to get a snapshot in time in the next week to understand, how many people have been infected in the last month, IgM might actually be a pretty good marker for that. But if you're getting towards the tail end of an epidemic, and as we're sort of hopefully going to be over the next month in the United States, we're going to really start pushing down cases. And then the real question is going to be, how do we open up the economy again? How do we get things moving? We really probably want to know more about the cumulative prevalence incidence that has happened over the whole duration of the epidemic. And so if it were me, I would choose IgG for that effort, to really be able to get a better understanding of just how many people have gotten it now that the actual epidemic curve is behind us. And that will allow us a lot more power to understand what to do next. So if I truly only had one, I think I'd always go with IgG for the most part. But luckily, if you can measure IgG, you can usually measure IgM as well. Let's see. Do you anticipate regional variation in antibodies, and how will this affect the response? This is a really great question, and it's a question that a lot of people who study antibodies right now are trying to understand more fundamentally about these questions. Are there genetic features of somebody's, does somebody's genome or maybe HLA dictate the type of antibody response they can produce? We know, for example, there's very good examples in the HIV field where there are people who are elite controllers, and there's been a lot of interest in sort of why some people can essentially become infected with HIV and not end up having very high antibody titers. And there's been a lot of interest and a lot of different theories and some data to show some of it is potentially antibody mediated. And so that might be genetic, for example, but there's a lot of interest to understand, are there genetic differences? And the reason I brought up genetics is because that oftentimes maps onto geography. And so we don't quite know with this virus yet if we're going to see those types of changes, but we anticipate that, there might be some. We'll have to see what ends up coming down in terms of data. So when will data on serological testing from CDC and others become available? It's a great question. I think in the next couple of weeks. What's that? What, Bill?
I'd really like to know the answer to that as well.
Yeah. So it's so CDC is saying, and the idea is to start a very large trial, a large study at the end of April to try to capture 100,000 samples from around the country in a representative manner to get some data. I think that it should be starting yesterday or a week ago, but it's unfortunately supposedly not starting until the end of April. But we're going to see before that a lot of studies are underway. The results are actually going to be coming out soon from different places. So my anticipation is we'll end up seeing some studies probably from Stanford and UW initially. We'll end up seeing some, and this is just US centric. Well, in Europe, I think we're already seeing studies that are coming out of Germany and Austria, the Netherlands. We'll probably have some data soon. Iceland is starting to have a different type of data. They're doing more surveillance for virus. And then in the US, in the next few weeks, I anticipate that we'll start having larger cross-sectional prevalence studies be performed, and the data will become available pretty quickly. So I think by the end of this month, we're going to have a much better idea of just how many people have actually had this virus. And by the end of the month, I think we will be able to actually start digging in and figuring out real policy towards opening back up the economy. And but until we get these first point prevalences, even if they're not perfect, then we need to hold on just a little bit longer before we start making really detailed plans about what's next. Let's see. Would you expect different levels of antibodies and potentially durability of immunity, if there is any, in people who had mild or barely symptomatic infection versus people who had serious disease? This is from Helen Branswell. So this is a really great question, and it's another one of these questions that we still don't know. And of course, these are not satisfying answers, but it is a novel virus. And so we don't know that, but we can look at we believe that there might be some some evidence, but it could go really both ways. If somebody has an asymptomatic infection, maybe that's because they were really able to ramp up antibodies quickly and be able to beat down the virus. On the other hand, if somebody has a very symptomatic infection, maybe they didn't get their antibodies up and running and their immune system up and running quick enough, but just that level of exposure might ultimately cause them to have an even higher IgG level later on. So this is a question that we're going to be asking and trying to understand, are there different titers that people have that are sort of helping us to understand what somebody's disease course might have looked like and vice versa. And it may or may not be important. We know, for example, when we measure the antibody titers, that's essentially the antibodies that have been pumped out from all of these plasma cells, which a lot of antibodies, IgG antibodies anyway, actually come from your bone marrow. The cells that make antibodies live in your bone marrow, at least the long-term ones oftentimes. And those antibodies, so some of those cells are more productive than others during a period of sort of quiescence or silence away from the infection. So we don't always know if a very high titer is even reflective of somebody being protective or not, because there's lots of other factors that go into your ability to fight off a virus. The antibody titer that's just hanging about in the blood all the time is one part of it, but some people might have very low antibody titers that just stick around the blood. But then upon exposure to an infection, they might actually have a really good capacity to spin up a lot of new cells and create new antibodies that wouldn't normally be sitting there all the time. So there's a lot about the immune system that we're still just trying to figure out, and these questions are really fundamental. So I think we're about to be out of time, so I'll answer this one last question. Can we talk about cross-reactivity? Can serology tests distinguish between SARS-CoV-2 and endemic strains? So a lot of effort goes into building appropriate serological tests in order to ensure that they can discriminate. So we definitely want tests that can be specific. We also want tests that can be sensitive. So you don't want it so specific that you end up capturing a very small fraction of the actual infected people. You want to hit some balance, but the good thing about antibodies is we can usually find some place along a virus's proteome or along all the pieces that a virus displays. We can usually find some part of it that is specific to that virus. Sometimes you actually have to be measuring multiple parts in order to get at it. But you usually can find good specificity. And in this case, we have one of the leading sort of candidates for a good antibody is the part of the virus that's on the spike protein where it actually binds to the receptor ACE2. And this is called the receptor binding domain. And this is it's very specific to SARS-CoV-2. There is some possibility that it could interact with SARS as well, but we don't really have SARS in the community at this point in time. And so in general, if we have an assay, in my opinion, that is finding SARS or SARS-CoV-2, I think that's pretty good. But receptor binding domain seems very specific, actually, and it seems pretty sensitive. So in that sense, we actually do have a molecule already that we can put into our ELISA tests and use that to be able to distinguish if somebody had an infection with one of the seasonal strains or with this new virus. But we're still undergoing a lot of validation. These tests will never be perfect, though. And one of the major reasons that these will not be perfect, in particular compared to PCR, is that PCR, you're kind of in good shape. You know that segment of the genome of the virus doesn't really change too frequently. And if it does, you can detect that through sequencing, and it usually won't change a lot. Maybe you have to update your PCR primers once every few months or once every few years or decades even. Antibodies are totally different. Every single person who has an antibody response will respond slightly different to different pathogens than any other person. We can even use people's antibody profiles to identify that person in the same way that we can use people's genomes to do it in a slightly different way, but it's possible. And so that just shows how plastic and how different people's antibody responses are, which means that leads to an immensely challenging problem to get every single to have a perfectly sensitive and a perfectly specific test. So there's always going to be some wiggle room there, and you're never going to have a perfect antibody test. But you can get pretty close because a lot of people do converge and develop antibodies, all of which look similar enough to other people's that we can actually create pretty universal tests. So I think I will leave it at that. I don't know if anybody really needs to chime in with anything. You can unmute yourself and say something, I suppose, but I feel like I just talked a lot, and I think we'll end the session there.